Okay. Oh, thank you everyone for attending. And thank you very much to Sam and the team at Syntica for all their help in hosting this webinar series. So just a little bit of housekeeping before we get started. We are going to keep the audio from all attendees muted throughout the presentation to keep down the background noise, but we are really keen for you to ask questions. We do encourage that. So please use the Q&A button on the Zoom meeting window and you can type your questions into that. At the end of the webinar, we'll have a question and answer session and we'll go through all of those questions. So as Sam said, I'm based in Auckland, New Zealand, and I have a PhD in physiology and electrical and electronic engineering from the University of Auckland. I officially started with telemetry research in 2009, but have been involved with the development of the telemetry system since it began 5 years prior to that in our lab at the University of Auckland. And I'm currently the Chief Scientific Officer at Kaha Sciences, but I also hold a 0.5 position at the University of Auckland as a Senior Research Fellow. And there my research interests include cardiovascular control, intracranial pressure, the development of implantable devices, and more recently optogenetics. And I use a lot of telemetry in our rat study, and I do some large animal non-telemetry work in sheep as well. So today I'm going to give you a quick run through of how carbon-based electrodes are used to measure tissue oxygen and the advantages of doing that and other studies by telemetry. I'm going to walk you through how we calibrate the tissue oxygen telemeters. And because our telemeters are reusable, I'm going to explain how you can remove, clean, and prepare those telemeters for reuse for your next animal. And then I'm going to give you some examples and show you how you can combine the tissue oxygen measurement with other pressure or biopotential signal measurements. So what do we mean by tissue oxygen measurement first off? Well, it's the measurement of the oxygen concentration in the extracellular fluid of a tissue. So as we all know, we have blood supply going to all of our tissues, and that breaks down into the capillary vessels that infiltrate all the tissues of our body. And those capillaries carry oxygen throughout the body, and that oxygen diffuses out into the extracellular fluid. Now, once it's in the extracellular fluid, it diffuses through and it's picked up by the different cells of that tissue, and it's used during the cellular processes. So the oxygen concentration in this extracellular fluid will be a balance between the supply coming in and the demand of the cells of that tissue. So when we measure a change in the tissue oxygen concentration, what we're doing is we could be detecting a change in either the local flow or perfusion of that tissue. We might be seeing changes in the systemic oxygen level, so if the animal is exposed to hypoxia or hyperoxia. Or on the other side of the equation, we might be looking at a change in the demand. Perhaps there's been a change in the neuronal activation and there's more oxygen being used, or some other change in the cell metabolism. So how do carbon-based electrodes actually measure tissue oxygen? Carbon-based electrodes were first used back in the 1950s, and their use in vivo work represents 15 years or more of research by Professor John Lowry and his team at the National University of Ireland in Maynooth. And we talk about carbon paste, what we mean is it's a mixture of graphite powder and a silicon oil. And how this works is that there's the reduction of oxygen on the surface of that carbon paste from the tissue it's exposed to or from the fluid it's exposed to. And that oxygen is reduced to hydrogen peroxide in the first instance, and then further reduced to water molecules. And you can see that that requires electrons. And that movement of electrons is measured as a current, which we can detect, because that current is then proportional to the amount of oxygen in that fluid or in that tissue. So the setup for recording tissue oxygen involves 3 different electrodes. We have the working electrode with the carbon paste at the tip and a reference electrode. And if we hold the electrical potential between those two electrodes at a set voltage, in this case minus 650 millivolts, we get a selective reduction of oxygen. We know that only oxygen will be reduced on that electrode. And then we have a third electrode called the auxiliary electrode. And if we measure the current between the auxiliary electrode and the working electrode, we can measure that flow of electrons that are used in that reduction of oxygen. And the equipment used to perform this is what we call a potentiostat. So the current between this auxiliary electrode and the working electrode will be proportional to the oxygen concentration in the tissue. Now, carbon-paste electrodes are more stable and are less susceptible to background noise than solid carbon electrodes, and that means that they can be more sensitive to changes in oxygen because you get a better signal-to-noise ratio. And there's also been shown to be minimal issues with protein deposits during chronic measurements. In fact, the team at Maynooth showed that oxygen measurement in the brain is stable for 12 weeks after the implantation. Now, this is in tethered animals, and the Kaha Sciences team, or the predecessors to that, the telemetry research team, got together with the team at Maynooth to develop a tissue oxygen telemeter. And so the tissue oxygen telemeter replaces that benchtop potentiostat to provide that set voltage between the carbon-paste electrode and the reference electrode and to measure the current. So there are 3 electrode leads on the telemeter. One of them, of course, has the carbon-paste electrode, and then we have 2 other platinum silver electrodes which act as the reference and the auxiliary electrode. So the working electrode, the carbon-paste electrode, obviously that needs to be placed where you want to measure the oxygen. It can't be used in blood, so we don't place it in a vessel, but it can be used in things such as the brain or in the kidney. And we do need to make sure that we fix the placement of those electrodes firmly in the tissue to stop them moving and causing noise on your signal. The reference electrode is placed nearby in a similar tissue, and then the auxiliary electrode is also placed nearby. And that needs to be a slightly larger surface area because that's what's providing the electrons for the whole process. Often that auxiliary electrode, if we're measuring brain tissue oxygen, is secured to a skull screw, and that will also act as part of the electrode. So the carbon-paste electrodes that we're using are 200 micron in diameter at the actual carbon-paste part. And this means that the volume of tissue that the carbon-paste electrode is sampling the oxygen from is a volume that extends around about 50 micron in all directions from that. So you can think of it as being a volume about 50 micron from the tip in depth and in about a cylinder shape, which is 300 micron in diameter. And so what you get is you get a measure of generalized tissue oxygen. And this means the signal is more stable than using a thinner electrode. If you use a thinner electrode, it's much more prone to changes from the individual cells or from the capillaries, and it's harder to get a stable signal and get a more generalized view of the oxygen. If you're interested in the details of how this system was validated as the telemetry system for measuring tissue oxygen, I'd like to point you to this article in the Journal of Neuroscience Methods. This is by David Russell. I'm going to show you some of the key points, but there is a lot more information in this publication if you're interested. So what David did was he implanted the telemeter in the abdomen of the rat and tunneled the electrodes to the skull. And you can place the carbon-paste electrode through a hole in the skull. The reference electrode's also placed a hole in the skull. And as I said, the auxiliary electrode in this case was attached to a screw in the skull. There's a couple of other screws here, and that's because they are used to anchor the dental cement and hold everything in place so you get a nice clean signal. And if we look at the average oxygen tissue oxygen across the 3 weeks post-implantation, you can see that after the signal settles, it's nice and stable during the 3 weeks following surgery. And there's no reason it wouldn't continue for longer and up to the 12 weeks or more that was shown with the tethered animals. So as part of the validation, it's important to see that the telemeter responds to changes in the level of tissue oxygen as well. And so in this case, the rat cage is placed in a chamber and the whole rat is exposed to different gases with different oxygen levels. And that's what's plotted on the right-hand side here. And the tissue oxygen in the brain is plotted against the left-hand axis. So you can see that baseline is about 21% oxygen in general room air. And then when you expose the animal to 100% oxygen, you get this increase in the tissue oxygen. And when the gas is returned to room air, you get a decrease back to baseline again. And if you also do it in the opposite direction and expose the animal to a 10% oxygen gas mixture, you get a decrease in the tissue oxygen before that bounces back to baseline once the gas is switched back to room air. So that's the exposure of the whole animal to a systemic change in oxygen, if you like. But the system also, or the electrodes also measure a response to a local change in the tissue oxygen. And in this case, the brain tissue oxygen is plotted against time after a IP injection of acetazolamide, which causes a selective increase in cerebral blood flow. And so you can see that straight after the injection, cerebral oxygen, tissue oxygen concentration increases because of the extra blood flow, and then it decreases over the next couple of hours. So Martin Kunis then took the telemetry system and looked at whether he could use it in his studies for measuring kidney tissue oxygen. And he's very interested in the role of the kidney in cardiovascular control. And what he found was that you needed, for the kidney, you needed a larger, or worked much better if you had a larger auxiliary electrode area. And so what he did was strip the Teflon off those platinum silver wire electrodes along quite a long length and coil it up to give a greater surface area. So the carbon paste electrode is placed at the site of interest to the depth that he's interested in recording at, and then the reference electrode is placed nearby with the auxiliary electrode coiled on the top and held in place by a surgical mesh and tissue adhesive. And of course, the body of the telemeter is still placed in the abdomen of the rat. And what Martin did was he compared the output or the measurement of the oxygen measured by the telemeter in anesthetized rats in the kidney to a standard Clark type electrode, which has traditionally been used for acute tissue oxygen measurements. And he found that there was a really good agreement between the tissue oxygen measured by telemetry versus the Clark type electrodes at a number of different depths through the kidney. And just like David Russell did in the brain, he also exposed the rats to changes in the oxygen, different gases. And so in this case, he's measuring medullary tissue in the kidney. And you can see that when the animal is exposed to 100% oxygen, the medullary tissue oxygen goes up. And then likewise, when it's exposed to a 10% oxygen, the medullary tissue oxygen goes down again and bounces back to baseline in between. So Tonya Imans in her PhD was working with Martin and others, and she used the telemeters for her studies, and she was looking at the difference in cortical and medullary tissue oxygen. And this is a graph from one of her papers from her PhD showing that particularly cortical tissue oxygen, but also medullary, has a circadian rhythm to that signal. And what's interesting is that during these dark shaded periods, or at nighttime, the active period for the rat, the tissue oxygen is higher than during the day. And they looked at the sodium excretion and urine output, and that did not follow the same circadian rhythm. And so what it's presumed is that this increase in tissue oxygen represents an increase in perfusion of the kidney in the dark periods or the active periods. And that makes sense because heart rate and blood pressure, cardiac output also show an increased level during those active periods versus the day and that circadian variation. But this just highlights one of the key advantages of using telemetry in that you can capture these variations across the day rather than having a single time point measurement if you had to do it manually in acute experiments. So Tonya also had a look at what happens when an animal is given a nitric oxide synthase inhibitor, L-name. And in this case, she used 3 different groups of animals. In one group, she measured mean arterial pressure or arterial pressure by telemetry in the abdominal aorta. And then in 2 other groups, she measured either cortical or medullary kidney tissue oxygen. And you can see that when the animal is exposed to nitric oxide synthase inhibition through L-name in the drinking water, you get an increase in mean arterial pressure as you would expect with the nitric oxide synthase being inhibited. What's interesting is that the cortical tissue oxygen in the black dots here, if anything, increases slightly towards the end with the big increase in mean arterial pressure, but the medullary tissue oxygen actually decreases. So she was able to tease out the differential response of the cortex versus the medulla in the kidney. So why would you want to measure any signals via telemetry? And I think the key advantage is that it enables you to measure your physiological variables in conscious, unrestrained animals. And this means that your results aren't confounded by the use of anesthesia, or they're not affected by handling stress of having to handle those animals before or during your measurements. You can also record from multiple animals running simultaneously in your laboratory. And I think most importantly is that it enables you to measure make long-term recordings that last over weeks or even months. And that's from animals living in their home cage undergoing normal behavior. So you can capture the long-term variation of physiological parameters and even during the development of a disease course over those weeks or months. And one of the key advantages is that telemeters are fully implantable, and so that lowers the risk of infection and improves your signal quality. The signal to noise ratio is very much improved because the amplifiers are right down inside the animal, and you no longer have this long wire of a tether picking up background noise or being affected by movement and creating movement artifacts on your data. Now, there is, of course, an initial investment in the telemetry system, but most users agree that this pays off very quickly because you're able to do fewer animals per group, and by doing a within animal comparison of before and after, or before and during your treatment, for example, with the development of a disease, you get a much higher statistical power, and that improves your results and the scientific questions that you can look at. And there's actually minimal day-to-day maintenance of the actual telemetry system. As long as you're looking after the animals and their welfare, the telemetry system will continue to record your data pretty much independently. So specifically the Kaha rat telemetry system, there are 3 parts to our telemetry system. We of course have the telemeters themselves that are implanted inside the animals, and they have different transducers and amplifiers to pick up the different signals that we're recording, and they transmit them to what we call the smart pad, which sits under the cage of each rat. And that smart pad, I'll go through in a minute, provides wireless power, but also acts as a receiver, and that's then connected to the data acquisition system. And our data acquisition system is very flexible. It can be any signal, any data acquisition system that takes in an analog input as the signal. And then the third part of the system is what we call a configurator system. It's software in this hardware box that is used to control the system. You only need one of those per laboratory, whereas you need a smart pad per telemeter or per rat cage that you're recording from. So we currently have 8 different telemeter models to choose from. We have some telemeters that have a Miller microtip pressure catheter on them, which is a solid state pressure sensor and gives a really nice high fidelity pressure signal. And we can also combine that with biopotential measurements of ECG, EEG, or EMG, for example, either with pressure or on its own. Or we have telemeters with a high gain amplifier for recording sympathetic nerve activity. We of course have the tissue oxygen telemeter that we're talking about today, and then we also have a new optogenetic stimulator and biopotential telemeter which we have just released this year. So the SmartPad and the ability to deliver wireless power to our telemeters is one of the key advantages of this system. The SmartPad uses electricity to generate a magnetic wireless power field, and that can pass through non-metallic surfaces such as the cage or indeed the rat's body. And then inside the telemeter, there's a pickup or inductor coil that takes that power from the magnetic field and converts it back into electricity, which is then used to power the electronics of the telemeter and to charge the battery. And this gives us 24 hours a day, 7 days a week power, and means that our experimental design is no longer limited by the battery life. It also means that we don't need for the telemeters to be sent back to us for refurbishment in between uses. Now, all our RAT telemeters have a built-in backup battery, and this allows for data collection away from the SmartPad. So the SmartPad can act as a receiver up to 5 meters away from the telemeter in terms of the data receiving. So if you wish to do something in a maze or in a chamber or somewhere where it's not appropriate to be recording while the cage is on the SmartPad, you get 3 to 6 hours, depending on the telemeter model, of battery life to be able to perform those experiments. And as I said, the SmartPad has analog outputs that can be connected to many data acquisition systems. And then that third and final part of the system is our configurator system. And this is key because it allows you to set your telemeters and SmartPads to communicate on one of our 40 independent transmission frequencies, which we call channels. And that means there's no data loss or interference of neighboring telemeters cross-talking with each other, and there's no need to shield your cages from one another. And it also allows us to perform diagnostics, check that battery level, all of those sorts of things as we go through our experiments. Now that's a bit of a whirlwind tour of our RAT telemetry system, but if you're interested in learning more, I encourage you to have a look at the webinar called Telemetry 101, Exploring Kaha Telemetry Systems, and that was presented by Dr. Phil Griffiths, my colleague, and the recordings are available on the Kaha website, and that goes into much more detail about the technology in our telemetry system. Okay, Sam, I'm going to pass it back to you now for the first poll, the next poll.
Thank you, Sarah Jane. And to give Sarah Jane just a quick break to catch her breath, I will launch the second poll. Mainly, we are interested in what parameter from a telemetry implant would be of most interest to you. Is it what we're discussing, the topic today, tissue oxygen? Perhaps is it ECG, EEG, EMG? Blood pressure is always a popular choice. Sympathetic nerve activity, temperature, activity. There's also a brand new telemeter from or parameter from Kaha Sciences, optogenetic stimulation. Give you a few more seconds to make your choices and give Sarah Jane just a quick chance to catch her breath. It makes sense that tissue oxygen is a very popular choice considering the topic. So it seems like the majority of you have responded. So I am gonna end this poll, and I will bring back Sarah Jane to finish her presentation.
Thanks very much, Sam. Okay, I'm now going to walk you through how we calibrate our tissue oxygen telemeters. So each of the carbon-based electrodes has an individual sensitivity to the amount of the oxygen concentration. And so they are individually calibrated after manufacturing at Kaha Sciences. And we do that by bubbling phosphate buffered saline, or PBS, with different gas mixtures. And that gives us a 3-point calibration. And so we've set up what's called a cell, which is essentially a small container with a tube for bubbling the gases into the PBS, the carbon paste electrode, auxiliary electrode, and the reference electrode. Now, we do this before the electrodes are attached to the telemeters, because obviously we're making replacement electrodes as well. So we use a benchtop potentiostat for this, and we can easily switch our gas mixtures using our system of taps here. And so if we look at the results when we use nitrogen bubbled through a solution will push out all of the oxygen and remove all of that oxygen from the solution. And so you get a very low current, around 0, in response to that. And then if we switch that to bubbling with room air using just a fish tank pump and bubble a solution with room air is about 21% oxygen in general. And so we get an increase in the current. Now, current here is plotted as a negative current. So we get an increase in that negative current. And you can see that it's quite variable, and it's also susceptible to changes in terms of just the bubbling of that solution. So what we do is we lift the tubing supplying the gas to the solution, and we get what we call a cloud reading. So that gas is still being pushed into the surface, to the surface, so it's not getting exposed to any room oxygen, but you're getting a nice stable measurement. And so we take that as our second value, and we then switch to measuring with 100%, bubbling the solution with 100% oxygen. Once that's stable, we again lift the tubing and get an O2 cloud, and that gives us our third and final measurement. And so we can plot the current measured by that particular electrode against the micromolar oxygen concentration that we know will be in those solutions after bubbling with that particular gas mixture. And the slope of this gives us the individual carbon paste electrode sensitivity, and that is reported on the packaging for both the telemeter with that particular carbon paste electrode, or if this was a spare electrode in the packaging of those electrodes. And just to point out that because we optimize the range of our telemeter to what you would expect to occur in tissue rather than in PBS solution bubbled at 100%, the use of the 100% oxygen would actually saturate if we did this attached to a telemeter. And so Dr. Pratik Thakur at the University of Auckland, who presented last week's webinar showing how he uses these telemeters in his research, what he does to get a third, he makes his own electrodes, and what he does to get a third calibration value is he uses a 10% oxygen gas mixture in here. So that slots between the nitrogen value and the room air of 21% value, and that enables him to do a 3-point calibration. The alternative is to do a multi-point calibration using just different mixtures of the PBS. And the way to achieve this is to take a known volume of PBS, in this case 20 mils, and add it to a glass cell with stirrer and electrodes, and we seal that container and saturate the solution with nitrogen for 30 minutes to drive out all of the oxygen. And then in a second set of, or solution of PBS, same known quantity, we then bubble that with 100% oxygen for more than 30 minutes. And so we know that there's a high oxygen, known oxygen concentration in that solution. And by adding very carefully calculated known volumes of the high oxygen solution to our original oxygen solution, we get these changes in the oxygen levels within that solution, and you can create a multi-point calibration chart that way. Now, these values are important because all of our telemeters and our smart pads know what the calibration values are set for each of the telemeter models. And so here, just to show you an example, I've recorded the SmartPad output without any calibration. Normally you wouldn't do this, you'd go straight to the appropriate calibration for that telemeter, but you can see that the SmartPad output in volts changes with a 3-point calibration that's occurring here. And by using the known calibration settings to change that voltage to relate to the current that the potentiostat in the telemeter is measuring, we can get the value of the nanoamp value of the current change with that different oxygen levels. And where the individual carbon paste electrode comes in is that then relates the current measured by the telemeter to the individual carbon paste electrode sensitivity. And so we can then divide that current by the sensitivity for that electrode, and that gives you the value of the tissue oxygen measurement in micromoles. So one of the key benefits of our telemetry system in the use of wireless power is that telemeters can be reused for multiple experiments. And so once you're finished with one experiment, you can carefully remove the telemeter from the rat, of course taking care not to cut those telemeter leads. You can then clean the telemeter with Tergozyme, which is an enzymatic cleaner. Now, most of our other telemeters, you would then just prepare the new electrodes or clean the pressure sensor and you'd be ready to go. But for the tissue oxygen telemeter, you do need to attach a new set of electrodes to those electrode leads. I'm going to show you the process of doing that in just a moment, but at the end of that process, you would then prepare the reference and auxiliary electrodes, stripping the Teflon off those electrode wires to expose the ends for your particular application, and you can then sterilize your telemeter for reimplantation. All of our other Kaha telemeters are suitable for ethylene oxide sterilization, but the carbon paste electrode of the tissue oxygen telemeter is not suitable for ETO. So most of our users use a 2% glutaraldehyde solution to perform that sterilization. And you can see here, this is an older telemeter with a different casing, but you can see that the electrodes are taped to the side of the container to keep them out of the glutaraldehyde solution. The telemeter and wires are soaked for at least 4 hours, or often overnight, before being rinsed with sterile saline just before surgery. And then these electrodes can be very carefully wiped with a 70% alcohol solution just to make them as sterile as possible, but taking great care not to damage the tip of that carbon paste electrode, because that will change the sensitivity or could even damage it beyond use. And if you're not using the telemeter straight away, the telemeter can simply be put on the SmartPad and checked with the configurator system to make sure it's fully charged before placing it in what we call safe mode, which is essentially turning the telemeter off, and it can be put away for storage until you are next ready to use it. So the process of attaching new electrode leads or electrodes to the electrode wires is relatively simple, but it does take some time. And you'll see why in just a minute. So the first step is to prepare the electrode leads, stretch the coiled electrode wire out, and remove the silicon, the colored silicon that fills the electrode tubing. And then you can insert a new electrode into the middle of that coiled electrode wire. And we use a silver conductive epoxy to make that join. And that does two things. It holds the wire in place with the epoxy once it cures, but it also acts as that conductive to form that conductive join. And because it's an epoxy, that needs to be left to cure overnight for a minimum of 12 hours. And we then coat each join with 2 coats of clear lacquer or nail polish, which involves a 2-hour drying time between each one. The joins are then covered with a small piece of polyurethane tubing, and then we coat all of that join with epoxy to insulate it from the tissue and the fluid. So that is again left to cure for overnight for a minimum of 12 hours. And we then test it for leaks by submerging the joins in PBS solution and recording the signal. Now, a high signal will suggest there's a leak, or we haven't quite got the epoxy covering that entire join, and so another layer of epoxy will need to be added and again left to cure. So you can see it takes a 2 to 3 day process of doing that. It's not difficult, but it does just take time because of the curing of the different epoxies. Then the last step is, of course, to remove the Teflon coating from the ends of the reference and auxiliary electrodes to prepare them for use. And if needed, to bend the end of the carbon paste electrode very carefully so you don't displace the carbon paste, so that it will be appropriate for its placement in the tissue. Now, measurement of tissue oxygen is quite an exciting advancement, but even more so if you can combine that measurement with a pressure or a biopotential signal to increase the number of the information that you're getting from your experiments. And we have facilitated this by what we call cohousing mode. And in this mode, one smart pad is able to optimize the wireless power requirements for 2 telemeters. And that can be 2 rats living in one cage, or it can be 1 rat implanted with 2 different telemeters. Now, the minimum animal size for 1 telemeter we recommend is 175 grams. So for 2 telemeters in 1 rat, we recommend the use of a rat that's 350 grams or larger, and they tolerate the 2 telemeters very, very well. And this enables you to mix and match between any of the 8 Kaha Sciences telemeters to get up to 4 signals or 2 additional signals to your tissue oxygen. Now, we still require 2 smart pads because 1 smart pad is providing the wireless power to both telemeters, but it's actually only recording the signal, receiving the signal from one of those telemeters. And we still need what we call a secondary smart pad to receive the signal from the other telemeter and to pass that to the data acquisition system. So this is just a quick movie to show you 2 rats living in one cage on a smart pad. And you can see the red and the blue line up the top. That's the arterial pressure measured in the abdominal aorta and the ECG from one of the rats. And then the green line down the bottom is the arterial pressure signal from the other rat. And you can see they're independent of each other. There's no crosstalk and no interference between those signals because the telemeters are operating and communicating on 2 individual different digital frequencies. And so this is just to show you how you would go about placing 2 telemeters in 1 rat. We place the telemeters in the abdomen by a midline incision through the skin and then separating the abdominal muscle layers. And we can use all of our telemeters on their hard case have these suture tab loops. And we use those to secure the telemeter to the abdominal muscle, and that keeps them in place, stops them moving around, and keeps them in the optimal place for the wireless power provided by the smart pads. As I said, you can mix and match your oxygen signal here with another telemeter. So you might choose to record tissue oxygen in the kidney with blood pressure and renal sympathetic nerve activity using a SNA telemeter, blood pressure SNA telemeter, and the tissue oxygen telemeter. Or you might choose to do what Pratik did in his the webinar he presented last week from his paper recently published in Stroke, and he measured tissue oxygen with intracranial pressure and mean arterial and blood pressure in the abdominal aorta using one of our dual pressure telemeters. And we can custom make these with a long catheter for recording from intracranial pressure from the brain. So you can see his setup here. He's got the carbon paste electrode, which is placed at his area of interest, the auxiliary and the reference, and then the ICP pressure catheter placed just subdurally through the skull. And this is just a short recording from the time of surgery when Pratik is causing a stroke in his animals using a mid-cerebral artery occlusion. So you can see that the tissue oxygen in the brain baseline is here, intracranial pressure and mean arterial pressure calculated from the blood pressure signal. And these grey shaded or blue shaded bars show you where the animal is anesthetized with isoflurane carried on 100% oxygen. So you can see the tissue oxygen in the brain increases. At the point of this red line, Pratik is inserting a suture thread up through the carotid artery to occlude the mid-cerebral artery, and that causes a sudden decrease in the tissue oxygen level of the area where the carbon-based electrode is measuring. And that's been carefully chosen to be in the penumbra, or the very edge of that stroke, or the tissue that's been occluded by that the occlusion of the mid-cerebral artery. The rat is then removed from the anesthesia, the suture's left in place, and it's returned to its home cage for 2 hours before it's re-anesthetized. You can see again the increase in tissue oxygen, but not as much as before, because the mid-cerebral artery has still been occluded. At this red line, the suture thread is removed and perfusion is returned to the brain, and you can see an increase in the tissue oxygen of the brain here. And now Pratik then followed these rats out through chronically through the development recovery from the stroke, and he looked at the blood pressure levels of those rats, and he looked at what treating the blood pressure to different levels, the effect that had on the animals, both brain perfusion and the tissue oxygen. So I encourage you to go back and have a look at the recording of his webinar if you're interested. And then in a similar study, this is an example from Pratik's supervisor, Dr. Fiona McBride, where she has used a similar setup with the tissue oxygen telemeter and a dual pressure telemeter to record intracranial pressure and arterial pressure in the abdomen. But in her case, she's also plotted cerebral perfusion pressure, and this is simply the calculation of arterial pressure less the intracranial pressure, showing you what the cerebral perfusion pressure, effective cerebral perfusion pressure, is for the brain. And Fiona also has an algorithm in her data acquisition system that allows her to pick up the respiration rate from the waveform of the blood pressure signal, and so she's been able to plot that here as well. And so you can see that if you measure the tissue oxygen in the brain, when you expose the animal in a chamber, much like we just showed you at the beginning of the webinar, to 5% carbon dioxide and 95% oxygen, a hypercapnia gas mixture, you get this increase in the tissue oxygen of the area of the brain being recorded. And you can also see that there's this increase in intracranial pressure, presumably due to vasodilation of the cerebral vasculature from the hypercapnia, and that's also showing contributing to this increase in the brain PO2 measurement. So just to finish up and recap some of the advantages of the tissue oxygen telemetry, it does operate on our 40 independent transmission and receiving channels or frequencies. The telemeters are reusable, so between uses you can clean your telemeter, attach new electrodes, re-sterilize your telemeter and implant it again. There's no need to send it back to us for refurbishment. The wireless power provided by the SmartPad enables 24-hour recording 7 days a week for the life of your experiment, and that can be weeks to even months. But that backup battery does give you the flexibility to record your data while the animal is away from the SmartPad for up to 3 to 6 hours, depending on the telemeter model. And that's ideal for doing behavioral studies. As I said, the flexible data acquisition, you can use the SmartPads can be connected to any data acquisition system that takes an analog input, and that gives you a wide choice of data acquisition. And the cohousing mode that we've just discussed gives you the flexibility to combine a tissue oxygen measurement with other telemeter models to record other signals that are important to your scientific questions. Okay, to finish up, I'd just like to point you to the Knowledge Centre section of the Kaha Sciences website. This is a password protected area of our website that contains a number of surgical videos, previous recordings of previous webinars, and a whole lot more information about our telemeters and their applications. That is free to access, but we do require individual registration for that. And so if you register, we will manually approve your registration. So it might just take a little while after your registration for us to do that approval, depending on the time of day and your time zone. But I encourage you to ask any questions of the in the Q&A question box. I see there's a couple coming through. But if you have any other questions, please do contact either Syntica or ourselves at Kaha Sciences on the email addresses provided. Okay, thanks, Sam.
Thank you, Sarah Jane. And as you mentioned, please ask any questions that you may have. We will do a transcript of all the questions that were asked with the answers and make it available in the coming days. So yeah, let's take this opportunity to answer the questions that have come in during your presentation. So the first one, Sarah Jane, that we got was some of the graphs show the oxygen signal plotted as millimeters of mercury rather than micromolar. Why is that?
Yeah, so this is, I guess, throwback to more traditional ways before you could measure the actual concentration in the fluid. And it's the oxygen content presented as what we call a partial pressure of oxygen. And the partial pressure of oxygen is a concept where in a mixture of gases, the total pressure is the sum of all the pressures in those gases. But if you think about a partial pressure as being the pressure an individual gas would represent if that gas was alone in the same volume. So that's a bit confusing, but if you think about pressure at 1 atmosphere or 760 millimeters of mercury, a 10% oxygen mixture would have a partial pressure of around 76 millimeters of mercury. And often we think of the rate of diffusion of gases being determined by that partial pressure rather than the concentration itself. Now, there are equations to calculate between partial pressure of oxygen and millimeters of mercury and the micromolar concentrations if you wish. But a lot of our customers, a lot of people tend to be using or presenting their data as a percentage of baseline or a change over time rather than those absolute units, because they find that that gives you a better power in their statistical calculations because of the variability in those signals.
That makes total sense. This next question is quite interesting as they ask, what other tissues has oxygen been measured in by telemetry?
Yeah, so all of the published work I've seen has been in the brain and then the kidney. I do know a group that tried to measure oxygen in cardiac tissue in the heart, but as you might expect, they found the signal to be quite noisy, probably due to the movement of that beating heart.
Too
And
fast.
it just wasn't feasible. And so we also did have a customer trialing the system to measure oxygen in tumors. And as far as I'm aware, they're having quite a bit of success with that. Unfortunately, that project was halted due to unrelated reasons. So they didn't ever complete that validation work. But I can't see why it wouldn't be appropriate for use. You just have to be able to place the 3 electrodes in that tissue that you're wanting to record the oxygen from.
Okay, good advice. Do you have to replace all the electrodes between uses, or could you reuse them if you remove them carefully?
Yeah, it's tempting to save time and perhaps just reuse them. The carbon paste electrode is actually quite sensitive, and so it definitely needs to be replaced between uses. You also get some infiltration of proteins and things like that into the carbon paste, so it's appropriate to replace them. And even the reference and auxiliary electrodes do get oxidized slightly during the process, and we certainly recommend that all 3 are replaced between each use. It's just if you're replacing the carbon paste electrode, then I think it's prudent to replace the others, because otherwise, you don't know at what point that's going to influence your signals.
Follow up on that question, how many times can the oxygen telemeter be reused?
Yeah, that's a good question, and the answer's a little bit variable because it depends primarily on how much you trim those electrode leads back between each use, and of course where you're trying to measure the their tissue oxygen. So the electrode leads are 28 centimeters long. And you can imagine that when you're inserting the length of the electrode into the end of the lead, you're probably trimming around about 6 millimeters at a minimum at each time you trim off your electrodes. And so you can imagine that the leads get slightly shorter each time you use them. So if you're measuring tissue oxygen in the brain, you're going to get a few less uses out of it than you would for the kidney, for example, because the body of the telemeter still has to be placed in the abdomen of the rat, and then the leads tunneled to your site of recording. So you do get multiple uses out of them still. They're plenty long enough, but you will get a few less for the brain than you do for the kidney.
Yeah, so it can change depending on the user and, like you said, the
Exactly.
leads, the length of the leads.
Yeah, how much you trim them back, all of those sorts
Right,
of things.
right. An attendee has an interesting question, actually. Can the telemeters be used to measure from a rat in a water maze?
Yes, it can. So that 5-meter range for the receiving is attenuated by water, but not to the degree that it can't be used. So you would need to get your smart pad closer than 5 meters, but we have done it in rats swimming in a water maze, and we found that we got really good signal reception if we placed the smart pad above the top of the water within 1 to 2 meters. We got really good signals, so you can certainly record from rats in a water maze.
That's pretty cool. Attendee asks about using the carbon-based electrodes or measuring, tissue oxygen in different animals. Am I correct in saying that right now it's just for rats?
Our telemeters certainly are the same theory, and a benchtop potentiostat can be used in other animals. Obviously, the size of the electrodes might determine how small you can go in which tissue, but the telemetry system that we have developed is specific for rats and perhaps guinea pigs but that would be dependent on the size of cages because of the wireless power provision to the telemeters by the smart pad.
Right, because one of the questions was in dogs, and I believe that's probably too large.
Yeah, you would be, it would kind of defeat the purpose of the telemetry system because you couldn't recharge the telemeter. So you'd be limited to 3 to 6 hours, which would get you nowhere really. So no, it's not suitable for telemetry in dogs.
Another question was asked about using this system in different stroke studies. Your thoughts?
It's certainly, yes, it's certainly possible, and it's very much appropriate to that. I would refer them to Pratik Thacker's paper or search for Fiona McBride stroke and rat stroke, and you'll find some papers, or have a look at the recording of Pratik's webinar and you'll see he presents some really nice data of the tissue oxygen measured in the brain in response to stroke and during the recovery afterwards.
Yeah, there was another question asked that I would also refer to that webinar and to that paper. They asked, does your research project project high blood pressure be protective against ischemic stroke? So to the attendee asking that questions, we also do refer you to that webinar we hosted last week, or to review Pratik's paper. There's another question that came in, and it asks why it deals with the sterilizing methods that you mentioned, and they ask why did you use 2% glutaraldehyde? I have trouble with pronouncing that one, but glutaraldehyde, and why not use ethanol or something else?
Yeah, so there are other options. Glutaraldehyde's not used everywhere. It's starting to be, some people don't like it. Ethanol is actually not a great chemical sterilant, but there are some other options. We just have to be a little bit careful because of the materials used in the production of the telemeters, but we do have a list of suitable other chemical sterilants available in our user manual, and we can point people to those rather than going through the list. They're all aldehydes similar to the glutaraldehyde, but ethanol is not actually a great sterilant for long-term implantation. It's good for cleaning and disinfecting, but it's not a sterilant as such.
Right, clear difference between sterilizing or just cleaning.
Yep.
Good point. And thank you. I think that's we've reached the 60-minute mark. But I'd like to thank you again, Sarah Jane, for your presentation. And if any others have any more questions, please feel free to continue to send them to us. And since we want to be respectful of everyone's time, we are going to wrap things up. We will be sure to answer any questions that we couldn't get to privately with anyone waiting for a response, and we will also publish a written transcript of all question and answers from our Q&A session. Again, I'd like to thank Sarah Jane for her insightful talk. And I am hopeful that all of you walked away satisfied that we met your learning objectives today, which were how are carbon-based electrodes used to measure tissue oxygen, calibration and preparation of tissue oxygen telemeters, and how combining tissue oxygen measurements with pressure and or biopotential signals. Please feel free to reach out to us or to Sarah Jane if you have any questions that you can think up of. As a reminder, our next session is titled Tips and Tricks for Successful Rat Telemetry Blood Pressure Recordings. And this is scheduled 3 weeks from today on Wednesday, December 2nd. And it will be presented by Dr. Phil Griffiths. He will mention and discuss an overview of the surgical approach for implanting Kaha Sciences pressure telemeters, how to identify and avoid the most common problems with abdominal implantation, how to ensure the best recovery of animal post-surgery, and understanding what unexpected signals in your pressure waveforms might mean and what to do about them. Thank you again for attending today's presentation, and we look forward to seeing you again at our next live event. Have a great day, everyone.